We know that bioinformatics has become an important tool to study the biology, to study the molecular biology. Now, in this module, I will try to illustrate that how we can use the bioinformatics to access and use the online data which is available to the scientists. Now, some very useful databases, they are kept at the National Center for Biological Information, which is abbreviated as NCBI. So NCBI is actually the guardian of that useful databases which is available to us. The online available data, which includes the whole genome, gene sequences, and protein sequences, these are available online to the scientists. And they all are being used in new researches and improved medical diagnostics. That is the advantage of this. For example, we will illustrate this statement with the example that imagine a physician is treating a patient with warts. And what he does, that he suspects that in the wart there is a viral infection, a viral cause, and even having a candidate virus, that is the papilloma virus, in mind. He thinks that there in the wart there is a papilloma virus there, and he excises the wart. He removes the wart from the patient, then homogenizes it in a buffer which contains a detergent to break open the virus particle. So he has some virus in the mind. He wants to confirm whether that wart contains that virus or not. So excises it, puts it in a buffer, and then in a detergent. Why? Because detergent break open the virus particle into DNA and the protein. Then he purifies the DNA and perform PCR with the primers specific for the candidate virus, that is the specific for the virus papilloma, which is in his mind. Now, all the study when he does on the SDS page, a band is obtained which confirms that the suspected viral DNA is present in the wart. So he obtains a band and he confirms that the DNA which is present is, as a matter of fact, it confirms like a papilloma virus over there. Now, to investigate further the exact strain of virus, that which virus is this, he sequences the part of DNA that he has amplified by PCR and obtain the sequencing gel over that and obtains the band. Just like this, a typical sequencing film where the sequences begin with CAAAA, CGG, and the rest of the sequence that can be read from the top of the film, which gives him a sequence. Now that is what he has done, the experiment. Now how he will search the sequence online. So to begin the search, he go to the NCBI database home page and click BLAST Nucleotide 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 BLAST, and then the BLAST N, and he types the sequence. That is the command he gives, that whichever he has found. Now, he adjusts the query range by centering figures in from and to range. That is from this range to that range. That will be the figures. For example, 20 to 200, 100 to 300, and things like this, which he puts in there. Then he selects the database in the present example. And in the present example, what he does, from the database, because generalized, he ignores mouse and human, as it is suspected to be the viral genome, and select others. So as he select the other command, he click the BLAST software program. The results, they are received in several forms. The computer gives him the result in the several form. First, as a colored bar graph that will be produced, which indicate the rough extent of the match between the query sequence and various sequences in the database. So what he does, when he puts the BLAST, the results are received in several forms, and a colored bar graph is received, which indicate the rough extent of match. That is the sequence he has discovered, and a rough extent of the match between that, and then the various other possible sequences, they will also be presented or projected in the database below the bar graph. So below the bar graph, the other suspected databases or sequences will also be presented, and below the bar graph that appear the sequences that match the query sequence. So it means by using the bioinformatics, he is sure that the virus he has studied is a papillovirus, and his study is further confirmed by the bioinformatics. So this is the advantage of using the computer and different databases which are present, which are available to the bioinformatic people.